Hi, my name is Angelo Chan, and in this video series, I will explain the HiC process and how HiC data is analyzed. The slides for this video can be downloaded in the links in the description below. Let us begin. I'll start by giving some background information on the topic before explaining what HiC is and the molecular biology behind it. Afterwards, I'll explain the bioinformatics pipeline for processing HiC data, first with an overview, and then elaborating on that at a higher level of detail. Let's start with some background. DNA usually exists in an uncondensed state. That is to say, it's usually a visually indistinguishable mass of DNA rather than condensed, distinct chromosomes. However, just because it's visually indistinguishable does not mean it's random. There are biological factors which affect the physical configuration of uncondensed DNA, and HiC is a technique for figuring out that configuration. Processed HiC data shares some similarity to linkage mapping data. It reports the frequency with which loci self-associate or associate with different loci. Loci association frequency can provide many biological insights, such as whether or not a locus is condensed, the presence or absence of certain proteins, and more. An overview of the complete HiC experiment workflow is as follows. We begin by obtaining our samples, which are either ready for immediate extraction or need to be cultivated first. Once the samples have been collected, they undergo the HiC Wetlab protocol, which produces chimeric HiC DNA fragments. The DNA is then sequenced, giving us raw reads in FASTQ format. These reads are then processed using a HiC bioinformatics pipeline to yield loci association data. This loci association data is then interpreted to give biological knowledge and insight. This presentation will cover the HiC Wetlab protocol and the HiC bioinformatics analysis part of this whole process. Now, in this presentation, I will only explain the parts of the Wetlab protocol required for a general understanding of the whole process, and only to the necessary level of detail. Wetlab procedures like preparing DNA fragments for next-gen sequencing have been omitted, as have the more meticulous levels of detail which are necessary for someone who intends to carry out the experiment. Situational bioinformatics steps, such as the conversion of raw sequencing output files to FASTQ files, are also not included. Everything in this video series is strictly HiC oriented. Another limitation to take note of is that there are many versions of the HiC Wetlab protocol and data analysis pipeline. There may be slight deviations between those and the process described in this video series. Also, it is assumed that the organism of interest is well annotated enough to have a standardized assembled genome. Additional steps will be needed when dealing with an organism which doesn't have a standardized assembled genome, and those steps won't be covered here either. So without further ado, let's go over the Wetlab procedures. We begin with a biological sample which contains DNA inside the cells, ready for harvesting. The DNA is cross-linked to proteins using formaldehyde. We now have DNA which is cross-linked to proteins. The DNA is then fragmented, typically by sonication or restriction enzymes. This gives us DNA fragments, some of which are cross-linked to proteins, and some of which are not. The unlinked DNA is filtered out, leaving us with only the DNA fragments which are cross-linked to proteins. Biotinylated nucleotides are then added to the ends of the DNA fragments, giving us biotin-tinted DNA fragments which are cross-linked to proteins. The DNA then undergoes ligation. Due to proximity, this almost exclusively occurs between either the two ends of a DNA fragment or the ends of DNA fragments which have cross-linked to the same protein. This forms a mix of chimeric DNA fragments and non-chimeric DNA fragments. The chimeric DNA fragments are formed from two originally separate pieces of DNA which were ligated together, while the non-chimeric DNA fragments are singular pieces of DNA which were not ligated to any other pieces of DNA. The DNA fragments then undergo exonucleation, whereby exposed nucleotides at the ends of the fragments are trimmed off. This includes the biotinylated tips. Our mix of chimeric and non-chimeric DNA fragments now have a key difference. The chimeric fragments have biotin attached to two of the nucleotides in the middle of the fragment, which were protected from the DNAs, while the non-chimeric fragments have no biotin. Keep this point in mind. The cross-linking is then reversed, giving us just DNA fragments. These DNA fragments then undergo biotin pull-down. Assuming 100% specificity, this should, in theory, leave us with only the chimeric DNA fragments, since again, in theory, only they should contain biotin. In reality, it won't be 100%, but that's the goal. These DNA fragments are then pair-end sequenced, giving us our HiC sequencing reads. Once we have the sequencing reads, it's time for the HiC analysis pipeline.